So, question 6. The classification of viruses is based on structure and nucleic acid types. Which of the following pairs of viruses both have an envelope? Right, so this is just something you're going to have to learn. It's just something you're going to have to learn. So you just have to memorize this. But if we go through it and write Ebola, which infected humans, lambdaphage, which infects, it's a virus that infects bacteria. And we have tobacco mosaic virus, which infects a tobacco plant. And we have the human immunodeficiency virus, which is HIV, which infects humans. So, here, Ebola would have a envelope. The phage doesn't, the TMV doesn't, and HIV does. So the answer is B. Right. Which of the following pairs of viruses both have a helical capsid? Once again, if we write it. Ebola does, lambdaphage doesn't, TMV does, and HIV doesn't. So the correct answer is A. Right, when a bacterial cell is infected with lambdaphage, the virus will enter the Lytic cycle. Describe the Lytic cycle of virus. So the virus has two cycles, Lytic cycle or the lysogenic cycle. The Lytic cycle is the active cycle. So here the virus particles are activated, so the DNA of the virus will be synthesized, yes? And so we're going to have the virus particles being shed. The lysogenic cycle, the virus does enter the host cell and does integrate its DNA or RNA into the host DNA. However, the virus particle will not be synthesized. So what will happen is it will remain in the host cell. That host cell will divide, so by mitosis, and we'll have more of the host cells being made. However, the host cells does contain the integrated virus DNA or RNA. So at the end, we're going to have a lot of host cells that have that the infectious agent DNA or RNA. So when it enters the Lytic cycle again, we will already have a lot of host cells with the virus DNA or RNA already present, so it just keeps on synthesizing large amounts. So the lysogenic cycle is dormant, whereas the Lytic cycle is the active. Right. So if we describe it, the first thing that happens is the virus particle attaches to the host cell. After it attaches, the virus particle will eject the genetic material into the cell. So this is penetration. The virus particle has effectively penetrated the host cell and has injected its DNA or RNA into the cell. So, after that, the virus particle will integrate its DNA or RNA into host cell DNA. And so what that means is it's gonna hijack the replicating mechanism of the host. And so what happens is the host will start to synthesize the genetic material and the protein of the virus. Why? Because this will enable a new virus particle to be made. And when you have a lot of the virus particles made and you've used up the cell and it's getting crowded, the host cell will lyse. And so you'll have new virus particles being distributed all out, and that will infect other host cells. And that's it. So, the multiplicity of infection is one factor that determines whether a virus enters the Lytic cycle or latency. A scientist needed to use a MOI of 0.5 for an investigation. The virus particles were at a concentration of 2 × 10 to the power of 9 centimeters per cubed, and the bacteria were at a concentration of 8 × 10 to the power of 8 centimeters cubed. Calculate the volume of virus particles that should be added to 0.25 centimeters cubed of bacteria. Right, so you probably haven't studied the multiplicity of infection, so reading this will throw the person off because you haven't studied it in biology, but instead they've thrown this at you in the question, and you're already nervous in the exam. So that's what Edexcel does. It will throw some new stuff at you, but the idea is they won't throw anything at you that you hopefully can't figure out yourself. What you're going to have to need is to stay calm and to break things up and to see what they're actually asking for and how you can get to it. And it's not important whether you know what a multiplicity infection is or not. You're just going to have to break it up so you know what you're timesing and dividing to try to calculate. But in the meantime, this just means the ratio between the virus particles that are present and the target cells, so that's the host cells that will be infected. That's just the ratio. So here we see calculate the volume of virus particles that should be added to 0.25 of bacteria. Well, that's the first thing we should start off with. Here they want 0.25 centimeters cubed, and if you remember your math skills, bacteria were at a concentration of 8 × 10 to the power of 8 centimeters per cubed. That per cubed, and this is just cubed, that means it's on 1 centimeters cubed. Right, so if we write it, so 1 centimeters cubed, and we want in 0.25 centimeters cubed, we divide by 4. And so if we divide by 4 over here, we're going to get 2 × 10 to the power 8. Right. So we've done the first bit of the calculation. Now, we can use this triangle because we have got enough information. We have the MOI, and now we've got the number of the target cells. So if we write this into a clearer triangle, that's the number of virus particles at the top, and in the left is the MOI, and on the right is the target cells that are present. So we have these two. So in order to get the virus particles, we just times it together. So we do 0.5 times 2 × 10 to the power of 8. And that should give us 1 × 10 to the power of 8. Now, we've got the number of virus particles. That's what we want. Well, no, we want the volume. Right, if you remember back to your maths or chemistry skills, you remember there's two ways to calculate volume. You can do D and V, or you can do C and V. Now, this is mass, and that's density. We don't have any of these, so that's unlikely we're going to use that equation. Now, another way to calculate volume, you have the concentration, and we have the number. Well, let's see, we do have the concentration present, because it's over here. The concentration of virus particles, that's one. And the number of infectious agents, well, we're going to be using the moles. So we have that too. So in order to get our volume, we do the number divided by the concentration. The number divided by the concentration. Now, that gives us 0.05 centimeters cubed, which is the answer. There we go.